Ever wondered how polarography works? Let's unravel this scientific mystery together. Polarography is a unique electrochemical technique that has piqued the interest of scientists worldwide. What makes it so intriguing, you ask? The answer lies in the core of its operation. Polarography measures the current that develops in a solution when a potential is applied to a unique component called a dropping mercury electrode. Picture this: a solution, an electrode, and a stream of current flowing because of the potential difference applied. Sounds like a science fiction movie, right? But it's not. It's a real-life practical technique used in laboratories around the globe. This technique has opened new doors in the field of electrochemistry, offering valuable insights and breakthroughs. Intrigued? Well, brace yourself as we delve deeper into the fascinating world of polarography. The journey ahead is filled with waves, potentials, equations, and much more. The theory of polarography is just as fascinating as the technique itself. At the heart of this technique lies a simple yet profound concept: the relationship between current and concentration of the analyte. So let's dive right into it. Polarography is an electrochemical analysis method. It involves the measurement of current that's formed when a potential difference is applied across a pair of electrodes immersed in a solution containing ions. The current that flows is directly proportional to the concentration of the analyte in the solution. This is the essence of the theory of polarography. But what happens at the molecular level? The analyte molecules, or ions, are reduced or oxidized at the working electrode. This reduction or oxidation is what we call a redox reaction. Now, when the potential of the working electrode is made progressively more negative or positive, the redox reactions start to occur at an increasing rate. This generates a current, which is then measured. The magnitude of this current gives us a measure of the concentration of the analyte. The more analyte there is, the more redox reactions there will be, and the greater the current. And there you have it, the basic premise of polarography, a direct relationship between current and concentration. But what about the shape of the current voltage curve, or the polarogram? Well, it's not a straight line. The curve is actually sigmoidal, or S-shaped. This is because the rate of the redox reactions is not only dependent on the potential of the working electrode, but also on the diffusion of the analyte molecules to the electrode surface. In other words, the rate at which the analyte molecules reach the electrode surface via diffusion also plays a crucial role in determining the current. This diffusion-controlled process is what gives the polarogram its characteristic sigmoidal shape. Now that we've grasped the theory, let's move on to the principles of polarography. The principles of polarography are the pillars that uphold this technique. With this in mind, let's delve into these core concepts that make polarography such a unique and powerful tool in the world of chemistry. Firstly, we have the half-wave potential, also known as the E1/2. This is the electrode potential at which the current is half of its maximum value in a polarographic wave. The half-wave potential is a characteristic value for every electroactive species, and it can be used as a fingerprint for its identification. So in essence, it's like a detective tool for chemists, helping them identify unknown substances with precision and ease. Next, we have the Ilkovich equation. This equation is the backbone of polarography, providing a quantitative relationship between the diffusion current and the concentration of the electroactive species. Named after the Slovak chemist Dionys Ilkovich, it is the equation that allows us to calculate the diffusion current in a dropping mercury electrode. The Ilkovich equation dictates that the diffusion current, id, is proportional to the square root of the drop time, t. It also factors in the concentration of the sample, c, the number of electrons involved in the redox reaction, n, and the molecular weight of the substance, m. So, to put it simply, it's a bit like a mathematical wizard helping chemists to calculate important values with accuracy. Now you may be asking, why are these principles so important? Well, the half-wave potential and the Ilkovich equation are critical in understanding and interpreting polarograms, which are the graphical results of a polarographic analysis. This means they're key to unlocking the wealth of information that polarography can provide. Now, with the principles of polarography understood, let's move on to the dropping mercury electrode. This is where the magic happens, and where we'll start to see the true power and versatility of polarography. So, stay tuned as we continue our journey into the fascinating world of this analytical chemistry technique. The dropping mercury electrode is a crucial component in polarography. It's the heart of the system, the engine that drives the analysis. This electrode, commonly known as DME, is unique because it constantly renews its surface with fresh mercury droplets. This ensures a clean and uncontaminated surface for each measurement, providing highly accurate and reproducible results. The mercury is released from a capillary tube, forming droplets that drop under the influence of gravity. As each droplet forms, it becomes the working electrode, the place where the magic of polarography takes place. Here, the analyte molecules are reduced or oxidized, giving us the valuable information we need about their concentration and identity. In addition to its self-cleaning feature, the DME also has a wide cathodic potential range, making it possible to study a broad spectrum of analytes. This dynamic, self-renewing electrode is truly the star of the polarography show. Next, we will explore the different types of polarography. Did you know there are different types of polarography? That's right, and today we're diving into the two main types, linear scan and differential pulse. Let's start with linear scan polarography, shall we? This technique is a bit like taking a leisurely walk across a landscape. But instead of hills and valleys, we're traversing voltage versus current. You see, in linear scan polarography, we gradually increase the voltage applied to the electrode at a constant rate. As the voltage rises, the current also increases, and we plot this relationship to form a polarogram. This type of polarography is straightforward and effective, much like our leisurely walk, but it does have a downside. The resolution isn't always the best because we're scanning everything in one smooth, continuous sweep. Now let's move on to the second type, differential pulse polarography. If linear scan is a leisurely walk, then differential pulse is more like hopping from one spot to another. Instead of a smooth, continuous increase, we apply a series of small voltage pulses to the electrode. After each pulse, we measure the current, and the difference in current between pulses gives us our data. This technique provides a higher resolution than linear scan because we're focusing on each spot individually instead of sweeping across the entire range. Each type of polarography has its unique features and benefits. Linear scan is simple and fast, making it ideal for initial investigations. On the other hand, differential pulse provides higher resolution, which is perfect when we need detailed information about the electrochemical behavior of a substance. So there you have it, the two main types of polarography, the steady linear scan and the detailed differential pulse. Both have their place in the world of electrochemical analysis, and choosing between them depends on what we need from our investigation. Understanding these types is essential, but it's also important to know where we can apply this technique. So let's move on to the next part of our journey, where we'll explore the various applications of polarography. Polarography has a broad range of applications. Let's dive into some of them. In the realm of chemistry, polarography is a hero. It's used to identify and quantify substances in solution, providing precise data on their concentration. You'll find it utilized in the analysis of organic and inorganic compounds, and it's particularly adept at detecting metal ions. Now, let's turn our attention to the medical field. Here, polarography is a valuable tool in clinical diagnostics. It's used to measure the levels of various substances in biological fluids, assisting in the diagnosis of numerous diseases. For example, it can detect abnormal levels of trace metals in the blood, which could indicate a health issue. And then we have environmental science. In this field, polarography is used to monitor pollution levels in bodies of water. It can detect trace amounts of heavy metals and other pollutants in water samples, providing crucial information for environmental protection efforts. But it doesn't stop there. Polarography also finds use in industries like food and beverage, where it helps ensure product quality, and in pharmaceuticals, where it aids in drug development and quality control. As you can see, polarography isn't just a technique for the lab, it's a technique for our world, playing a vital role in many different areas. Polarography, as we've seen, is a versatile technique with a wide range of applications. We've covered a lot today, haven't we? We've journeyed from understanding the basics of polarography, delved into its theory and principles, and explored the dropping mercury electrode. We've also looked at the different types of polarography, namely linear scan and differential pulse. Finally, we touched upon the real-world applications of polarography. It's been a packed session, full of insights, Subscribe for more because you won't find me later. Until next time, keep exploring the fascinating world of chemistry.